Abdulnasser. Our experiment was about targeting an RNA binding protein that's related to the development of breast cancer, which is non-PO domain counting octamer binding protein, also called NONO protein. As we can conclude from these pie charts done by WHO, we can find that breast cancer is the most common type of cancer with an incidence of 21.9%, including all ages of both genders, while it accounts for 38.8% in female cancer. Breast cancer death in the UAE has reached up to 16.6% despite chemotherapy, so targeted therapies are urgently needed. We have found that NONO protein is overexpressed in many types of cancer cells, especially in breast cancer, as it binds to the RNA and activates the proliferation of these cancer cells so that they will multiply and develop excessively. For NONO protein to be activated, it has to be methylated by protein arginine methyltransferase 1 and 4, and it has to be phosphorylated by cyclin-dependent kinase enzymes. So our aim for this experiment was to inhibit NONO protein and thereby achieve our goal, which is treating breast cancer. Methylation was inhibited using PRNT inhibitor, which is ADOX diluted in DMSO solution, while phosphorylation was inhibited using CDK inhibitor, which is roscovitin.
Regarding the methods, in our experiment we have used two cell lines, MCF7 and MDAMB231, which were seeded in 6 wells plate, with a density of 10 to the power of 6 cells per well. The next day, we added 20 micromolar of ADOX or roscovitin in each well and incubated the plates for different timings. With ADOX, we incubated the plates for 0, 6, 12, 24, and 48 hours, while with roscovitin, we incubated the plates for 0, 12, 24, 48, and 96 hours. We also did control wells by adding only the vehicle DMSO. At every time interval, cell viability and proliferation were assessed. For the cell viability test, cells were washed with PBS, then trypsin ETTA was added to deattach the cells in the incubator, which was set at 37 degrees Celsius and 5% carbon dioxide for 3 to 5 minutes. After that, cells were counted using an inverted microscope and a hemocytometer. So trypan blue, which is a dye that only stains dead cells, was added and counted the cells that are negative for trypan blue, which are viable cells, and also cells that are positive for trypan blue, which are dead cells. To get the viability percentage, we used an equation, which is the number of viable cells over total number of cells times 100. For the proliferation test, we counted the viable cells at every incubation period and compared them to the number of cells in the control well. For example, DMSO at 6 hours, there were 10 to the power of 6 cells, which represent 100%. Then, ADOX at 6 hours, there were 0.5 times 10 to the power of 6 cells, and by cross-multiplication, we got the percentage to be 50%. ADOX and roscovitin come in powder form. Therefore, to change them to liquid form, a vehicle must be added. And in our experiment, we have used DMSO. Therefore, we have done both experiments with only DMSO and without the inhibitors to check if the results obtained were due to the inhibitors. From the graphs, we can see that the results are due to the inhibitors since the proliferation and viability percentages remained almost the same at every incubation period.
Discussing the results, let us get to know the differences between the two cell lines used in the experiment and why are the results different but lead to the same conclusion. MCF7 are estrogen receptor positive cell line, which is the mechanism of growth of these breast cancer cells. They have receptors on the cell surface that allow the binding of the estrogen to fuel the cancer cell division and growth, and this estrogen is taken from the bloodstream. MDA-MB231 is a triple negative breast cancer cell line, which means it lacks the estrogen receptors, progesterone receptors, and HER2, which are usually targeted for killing cancer cells. Because it lacks all of these receptors, it is known to be the most aggressive, most difficult to treat through conventional ways, more likely to spread among diagnosed patients, and more likely to come back after cancer treatment. The treatment options are fewer than the MCF7 treatment options. This puts pharmacists on a mission to find treatment options for MCF7 breast cancer cells and the most aggressive ones, MDA and B231. And we can say that we have found a lead to our target through inhibiting NONO. As seen in the graphs, there's a reduction in the proliferation and the viability in both cell lines, which proves the efficacy of our strategy of inhibiting the methylation and the phosphorylation processes of NONO. But if we compare the two cell lines, the reduction is higher in the MCF7 than the MDA and B231, because as explained earlier, it is more difficult to treat. However, our strategy was still successful at reducing the proliferation and the viability for both cell lines, including the most aggressive ones, MDA and B231. This means our work does not stop there. In the future, we as Ain University pharmacy students will work on inhibiting NONO, then evaluating the cancer cell proliferation and viability, analyzing methyl NONO and phospho-NONO in cancer cells versus normal cells, inhibiting PRMT, then evaluating methyl NONO, inhibiting CDK, then evaluating phospho-NONO.